So are there any questions or comments on Carl's presentation?
Very interesting. As you showed me in New Orleans, I think, the RNFL pictures you're playing with. In glaucoma, we have a big group of patients. We reach the floor, and that's all. That's OCT stops. What we are measuring is the thickness of the RNFL. Looks a little bit primitive now with all these pictures. Are you doing something for that? At that floor effect, you still can count some RNFL that's hiding inside the structures?
I think that you'd be the best person to answer that.
There will be an interesting point to study, because nearly 10–20% of advanced glaucoma patients, we have to go back to the visual fields. I want one day to get rid of the visual fields with the OCT.
Yeah, it's a very interesting question and also a difficult one. Now, I didn't show thickness maps and the like in my presentation, but of course all of those quantifications, the AP study, the RDB, are based upon detailed thickness analyses. One of our scan modes is the 12:9 wide scan, so fairly broad scanning area and range. And the RNFL can be assessed over that whole range. And of course, clinically, the circumpapillary RNFL is used most often, but you, as a user of the device, the data is available to look at in a much more detailed manner than simply the CP-RNFL quadrants or clock hours and so forth. And I'll also comment that with respect to glaucoma, in the Triton US software that I showed there, the disc analyses are far improved, especially the cup measurements for normal subjects, and it really makes a significant difference for glaucoma discrimination.
You want to add to that, Charles? Sure. Don Hood, Columbia University. I'll be talking tomorrow about a report that we've developed with Topcon. And one of the things that may be surprising to the retina experts is, by and large, the glaucoma experts don't look at the scan. So the answer, part of the answer to the question is the segmentation in that machine is excellent. But part of the answer is if you actually look at the circumpapillary scan image, and one of the principles we'll be pushing is that the report should have an image that's big enough to look at, you will see in some places there is no residual. Even though I'm part of a group that pushed that there was a residual, you could actually see local defects and look at it the same way a radiologist would look at a tumor in your brain, as opposed to believing these summary numbers.
Very good presentations. With regards to uveitis, currently we use a lot of subjective measures of disease activity, such as vitreous haze. Are there any efforts to utilize the swept source OCT to come up with more objective measures of disease activity, such as vitreous cell counts or anterior chamber cell counts?
There are several groups that are working on that at this moment to try to get an objective vitreous cell count. So yes, my hope is in the future that we can get objective uveitis measurements, because they're important on one hand for patient control, but also they're important for the pharmaceutical studies. Because as said, if I ask 5 of my residents to give me an evaluation of uveitis, I get 6 different results. So we need a much more objective way of doing it. So yes.
I think one comment on that is that it's hard to measure absolute signal, right? Because you have cataracts, you have a lot of confounding issues. If you actually have a cell count or something that is not a pure signal or reflection measurement, that'll be more reproducible.
Yes, yes. So we're focusing on cell count, not on haze, because trying to visualize and quantify haze, while potentially possible, is proving to be very challenging. Because you'd have to find some way of normalizing every single scan you do, because even in the same person, the same scan can be different if you do it 10 minutes apart, and the absolute signal can be changed. So we need to find a way of normalizing that. So I think that's something for the far future. But in the closer future, what I showed is that the cell count itself is in the data. And you can use it inside the data. Now it's just up to us and the researchers we're working together with to come up with a very stable way of counting those cells.
So I had a topic that I wanted to just briefly discuss. So in the morning session, we heard that there's potential to reduce the cost of OCT technology dramatically, okay? But the problem with this is that that assumes that you can make the market very big. Okay, so if you say, well, the market is like the cell phone market, or self-driving cars, for example, use laser radar, which is kind of analogous to OCT, but there's millions, billions of cars, right? The ophthalmology market is very limited. And so the question for this panel that I'd like to ask is, what is your assessment of expanding the market? How does the market size, the business model trade off against cost? Do you see OCT in the home environment, in primary care, in high-risk populations, or are we going to be limited in size in the near future?
Well, certainly, we hear practically all the time now about potential home use of OCT devices or moving OCT into the general practitioner's environment. So I do think we're moving in the direction of higher volume. That said, I don't think we're ever going to get to the volume of cell phones and telecommunication systems. So I don't know if this market is necessarily going to drive down the cost in that manner. But we should also consider that there's a lot of clinical value, even in the short to midterm, for these higher speed technologies. As Professor Fujimoto showed, the higher speed, enables techniques like VISTA. On a much more simpler scale, it enables larger field OCT angiography scanning. And looking at it in the inverse, it could potentially provide far more averaging and then effective resolution for much smaller area scans to get better visualization of, say, choriocapillaris structures. So I think there's a lot of potential clinical value, even in the short to midterm, before we even think on the scale of driving down cost to telecommunications type level.
From a clinical point of view, I would say that it's definitely where the future is going to go, is to have a low-cost, small unit that you can actually be using at home, but that needs to be able to do the diagnosis itself to a certain degree, or at least say, not make a diagnosis, but say pathological or not pathological, and then bring the patient to the next level of care. And I think if this happens, I think that it will revolutionize the way we do medicine, because we're going to catch diseases at a much, much earlier stage and be able to treat them in a much earlier stage than we do now. And in that sense, I do think it is where the future is going, but it's not going to be anytime soon.